Today I'm going to present our lab's work on quantifying the impact of non-coding variants on TF-DNA binding. So we all know that genetic variants can play a very important role in the pathogenesis of many complex diseases. If these genetic variants occur in the coding regions, we have, can you see the title? So if these variants happen in the coding regions, there are established measures that show the functional impact on genes. But however, many recent studies have shown that the majority of the disease-associated variants are actually in non-coding regions, and it is still difficult to quantify the functional impact of these non-coding variants. There are various ways a non-coding variant can affect gene expression. So in our lab, we are particularly interested on the impact on TF-DNA binding. So basically, the non-coding variants can disrupt the interactions between TFs and their DNA binding sites. And in this talk, we will focus on a very common and specific type of non-coding variant, which is a single nucleotide variants, also short-named as SNVs. So there are some current methods that try to predict the binding change due to SNVs. Most of these methods are based on PWM, which is position weight matrix. They are just the matrix behind these very popular motif logos. Basically, given a PWM, we can score a sequence, and if we want to study the difference between two sequences, we can simply take the difference between the two scores. But if we look at the score itself, we want to ask, is this difference significant? There is no very direct way to derive a statistical distribution for this difference. So if we use PWM as the model, it's hard to answer this question. The other thing is, if we look at the calculation more carefully, we can see that actually only one base is being used, which means we are not actually taking into account the context of the mutation. But this is a very questionable thing about PWM because it assumes the independence between bases. So ideally, we want to build a model that can give us the distribution of the difference we predict. Also, we want to use the information on the sequence context of the mutation. So in practice, we have found that PWM-based methods, generally, they can detect large changes in TF binding, for example, from a high affinity binding site to a non-specific binding site. But they ignore less drastic changes, which can also have important phenotypic effects. Okay, now we start to build our own models. And the raw data we are using are universal protein binding microarray assay. So this is a high throughput in vitro assay. It is TF specific. In each experiment, we can have a quantitative measurement of the binding specificity of about 40,000 DNA sequences. The design of the sequence on the microarray is very balanced, so that each temer appears once and only once. We can find this data on online databases such as UniProbe and CISBP. On the left, you can see an example of UPBM data. We have the signals and also the genomic sequence. So this raw data, we can't directly use them, so we need to process them. What we do is we count the number of consecutive k-mers and build a design matrix. For the outcome variable, we simply take a log so that the error terms turns out to be approximately normally distributed. So yeah, this is an example taking k equals 2. which means we use dimers. And since we have this matrix, it is quite straightforward that we can build a linear model. There's nothing fancy about this model. Just one thing I want to point out is the number of features will grow exponentially with the k. So as k gets larger, we have larger dimension for the design matrix, and the design matrix also gets more sparse. Since we have the model, we want to get the estimate for all the coefficients. So this is like we use the OLS estimate. This may sound like an old method, but it actually has some benefit. It gives us unbiased estimate of the coefficients in the linear model. But more importantly, because we previously discussed that we want the distribution of the estimates. So OLS in this sense is a good estimator. So there is one thing I need to point out is we have a perfect collinearity due to we are using fixed sequence length, which means if we add up all the counts of dimers, they will add up to the same number, and this column would be collinear with the intercept column. That means the individual betas are not estimable in the statistical sense. However, the linear combination of the betas are estimable as long as the lambda is in the row space of the design matrix. And for any estimable functions, we can derive the distribution of the function. So why are we interested in the linear combination, not individual betas? It's because our target is to predict the binding change. And here is a case like if we have a mutation in the genome, and we assume that k equals, we use a 6-mer model here. So there are 6 6-mers that are being affected, overlapping this mutation. So the binding change in our model is a linear combination of the corresponding betas. And we have shown mathematically that any lambda that represents the sequence change due to a SNF is in the row space of X. And that means all the binding change is estimable. And here on the bottom is a simple output of our OLS estimate. So we are given a reference sequence, the mutated sequence, and we can calculate the difference as well as a t-statistic. With regard to that difference, we can also give a p-value. There's one thing more to confirm is the best k to use. So we started off like showing a 2-mer model, but that was like too simple. So we increased k, but when k gets larger than 6, we can see that if we use independent testing data, a signal of overfitting kind of shows. So in our research, currently we use 6-mer models. Okay, now that we have the model, we want to assess whether our models can predict the phenotypic effects. So this first part, we are asking whether the changes in TF binding can predict the changes in gene expression. And this part of work is done by Dora Li. So to get the expression data, we are using an assay called Massively Parallel Reporter Assay, published in Nature Biotech in 2012. And basically, this is on the top, it is a synthetic enhancer that contains 4 binding sites for the transcription factor CRAB. They did this for a lot of TFs, but we, as a case study, only use CRAB. So in this MPRA, they introduce all possible single base pair mutation to this 87-mer sequence, and also some multiple random mutations. And they can measure the gene expression in this way for the original sequence as well as each mutated sequence. So what we want to get from this MPRA is the expression change. So it is on the y-axis, and we use our model to predict the binding change, and we want to see the correlation between the binding change and expression change. On the left is the prediction using PWM models. On the right is our model. So firstly, we look at the CREB number 4, the putative binding site that is most close to the transcription starting site. So we can see that for this site, both our model and the PWM model predicts pretty well, but our model predicts like a little bit better than it. So we kind of take out the same analysis for all the 4 putative CREB binding sites, and we see that although our method doesn't consistently perform better than the PWM, when the PWM predicts good, we predict even better than them. And I want to point out that CREB is may not use all of these 4 binding sites, because most likely it is using the closest one to the TSS. So that means our model in general is predicting better than the PWM model. Next, we are looking at multiple mutations. So now it's not a single mutation, but 2 or more mutations in the putative binding sites. And also we can see that generally speaking, we are doing better than PWM. And what our model does even better is if we look at a longer region. So here we look at CREB number 2 and number 3, these 2 binding sites, as well as the parts between them. And we carry out the similar analysis, and we get an R-square of 0.49, while PWM got a 0.25. And similarly, looking at longer regions, I'll just be quickly. So here is, if we look at all the mutations in this sequence, and we predict much better than PWM. And we also want to point out that the MPRA replicates gives us an R-square of 0.66. That's kind of like an upper limit of R-square we can get. Another way to assess whether our models can predict the phenotypic effects is through applying our model to predict the effects of pathogenic SNPs on TF binding, and this work is carried out by Zhang. So what we did is we chose non-coding pathogenic SNPs annotated in the HGMD and ClinVar database, and we select the controls from the 1,000 genomes. We used all the ppm data available on CISBP and UniProbe for human and mouse TFs. And for each SNP, we kind of calculate all more than 900 predict z-scores, and we use the maximum absolute value of it to use as a measure of the binding change due to that SNP. And if we look at the distribution, if we compare the distribution of the pathogenic SNP and the control SNP, we can see that the binding changes caused by the pathogenic variants are significantly larger than the changes caused by the control variants. Okay, last, a summary. So I want to just emphasize that the novelty of our approach is we can not only compute the change in TF binding, but we can also compute the distribution for that change, which means we can get a z-score as well as a p-value for the change. And in the future, we are looking forward to replace the OLS estimate by some Bayesian method, so that we can apply it to even 9 more or 10 more, which means higher dimensions. With that, I want to thank my PI, Raluca Golden, and all the members that participate in this research. Thank you.
So you showed the comparison to the PWM. So where was the PWM from? Is it downloaded from the database, or did you train it yourself?
We downloaded from the Jasper database.
Ah, okay. So this may be, some PWMs may be more accurate than others. You know, there was this competition between different methods to like
we actually also, do you mean that if we derive the PWM from the PBM data we have?
No, I mean if you use a different algorithm to derive it, like BML PBM or feature reduce in one of the...
I think that's a good point. We should use more PWM derivation models.
In like more accurate.
Yeah, and we'll give more comparisons in the future.
Yeah, and also I was wondering, so you showed the comparison of different K values on PBM data, but did you also compare them on the reporter assay that you tested on?
Oh, that's a very good point. Yeah, maybe in the future we'll train, although it doesn't predict well for the independent PBM, it may predict well for...
Yeah, it could be 5 mers better than 6 mers on the reporter assay.
Yeah, thank you for the suggestion.
Let's thank Jingkang again, and we move to the next speaker.